Greetings, friends. Welcome back. Welcome back for anatomical analogies. Not going to be as many analogies here with some of these discussions, but we'll try to slide them in there as much as we can. But mostly just going to be spitting facts, spitting fire, spitting info, spitting knowledge for us to really get it up in the brain there, so it sticks. There. So today, in this episode, we're going to discuss DNA. So DNA, as we mentioned prior, they're composed of nucleotides. So a bunch of nucleotides bind together to form a nucleic acid. So there's the DNA and then the RNA. We're going to discuss the DNA in this video here. So as we recall, nucleotide is a pentose sugar attached to a phosphate group and also attached to a nitrogenous base. So we go a little bit deeper in this discussion here. So the pentose sugar with a DNA is deoxyribose. So deoxyribose, it's a 5-carbon sugar. Just remember deoxyribose DNA. So deoxyribose DNA, it has that D to start each of them. Attached to that is a phosphate group, and attached to that pentose sugar, that deoxyribose, is the nitrogenous base, except the nitrogenous base always with DNA is going to be adenine, guanine, cytosine, and thymine. So nucleotides pair with other nucleotides to form the nucleic acid, as we already know. DNA, it must be double-stranded. That's a major differentiation factor between DNA and RNA, is they're double-stranded, and as you recall, since they're double-stranded, DNA are too large to leave that nucleus in the formation of proteins. But we already discussed that. But nitrogenous base, as we just touched on, adenine, guanine, thymine, and cytosine. So the adenine always binds to the nitrogenous base of another nucleotide that is the thymine. So the A and T bind together. There, so it's the pentose sugar, the phosphate group, and then so let's say adenine, that's one strand. On the other side of the strand would be deoxyribose, phosphate group, and then the nitrogenous base would be thymine, because A and T bind together, so adenine and thymine always bind together. So if there's a T over here, there's an A over here in this strand. If there's let's say a cytosine here, there's going to be a guanine over here. So C and G bind together, or G and C bind together, whatever, it doesn't matter. This could be a C, this could be a G, or this could be an A. If this is an A, this has to be a T. If this is a T, this has to be an A. If this is a G strand of the nitrogenous base, this has to be the cytosine of the nitrogenous base there. So on it's gonna, all these strands of each nucleotide are gonna bind together. So let's look at it like this here. So this is a rough little drafter. So this is the phosphate group, this is the sugar, this is an adenine, and obviously on this side, to reflect it, that this single strand, since DNA is double strand, it's going to have a whole complete other side. So it's going to have the thymine there attached to the deoxyribose attached to the phosphate group. So this whole chain here, this is a nucleic acid, this is a nucleotide, this is a nucleotide, this is a nucleotide, this is a nucleotide. And it's kind of like a ladder if you think of it that way. So each strand, each side is going to be the linear portion of the ladder, and then the combining the A and the T, or the C and the G, or the G and the C, or the T and the A, the adenine, the thymine, cytosine, and guanine binding together is going to create the stools of the ladder there. So DNA, double-stranded, it has the pentose sugar, which is deoxyribose, attached to the phosphate group, which is and the deoxyribose is also attached to the nitrogenous base, which is always with DNA. It's either adenine, guanine, cytosine, or thymine. Adenine and thymine are complementary pairs, and cytosine and guanine are also complementary pairs there. So, excuse me, that's DNA. Now we're going to look at RNA. So there's some similarities, but there's definitely some differences here.